Bismillahirrahmanirrahim. Assalamu alaikum student. Today we are going to discuss with the histology of the female genital system. Female genital system consists of the pair of the primary sex gland, the system of the genital ducts and the external genitalia. So today we are going to discuss with the histological features of the different parts of the female genital system, including the ovaries, the oviducts, then the uterus, and then the vagina, cervix, and mammary gland. So what about the details of the female genital system? It consists of internal genitalia and external genitalia. In internal genitalia we have paired ovaries, we have paired oviducts, then the uterus, and then the vagina. And in external genitalia we are having labia majora, labia minora, and the ovaries, both acting as an exocrine and endocrine function. As exocrine, they are producing the gametes by the process of gametogenesis, and as endocrine, they are producing the estrogen and progesterone hormone. So in nulliparous women, the ovaries are the almond shaped pinkish white in color. They have a size of 3 into 1.5 into 1 centimeter. In case of the multiparous women, they are scarred and irregular because of the frequent ovulation. The ovaries have a superior pole and they have an inferior pole. By means of superior pole, with the help of the suspensory ligament, they are attached to the pelvic walls, and at inferior pole with the ovarian ligament, they are attached to the uterus. Histologically, ovary composed of cortex and medulla, and cortex is covered by the outermost layer which is called germinal epithelium. This word germinal epithelium is also a misnomer because previously it was thought that the germinal cells, that is the gametocytes, are derived from the germinal epithelium. Now we know from the knowledge of embryology that these germinal cells, oocytes, are derived from the epiblast, move to the area of the yolk sac, and then they migrate from the yolk sac to the area of the developing gonads. So this germinal epithelium word is a misnomer. Histologically, this layer is simple cuboidal epithelium. Just beneath the germinal epithelium, there is a connective tissue layer called tunica albuginea, and beneath this tunica albuginea, there is a cortex where we have a pool of the oocyte in different stages of development. And innermost part of the ovary consists of the medulla, which is loose connective tissue containing vascular bed and nervous tissue. Ovarian cycle consists of the three phases: the follicular phase, the ovulatory phase, and the luteal phase. And in follicular phase, there is a development of the primordial follicle that develops into mature follicle, and this is the follicular phase of the endometrium. In ovulatory phase, there is release of the mature follicle, and in luteal phase, the residual follicular cells folds and become part of the corpus luteum. There is a secretion or luteal phase of the endometrium in which progesterone is secreted. So first we will study the follicular phase. In follicular phase, you can see this one is the primordial follicle, and primordial follicles are formed in the female in the prenatal life. They consist of the primary oocyte, which is arrested at prophase 1 of the first meiotic division, and this primary oocyte is surrounded by the single layer of the flat follicular epithelial cells. At the time of the puberty, under the effect of the different hormone like follicle stimulating hormone, some of the primordial follicles are recruited and then they start the growth. Their epithelium initially become the simple cuboidal, and then there is stratification of the epithelium. At the same time, the zona pellucida is also secreted, which separates the primary oocyte from the surrounding stratified granulosa cells. In the secondary oocyte, the antral spaces starts to appear and the lichar folliculi is formed, and when these antral spaces coalesce to form a large crescent shaped cavity, the follicle is given the name of the Graafian follicle. Histological slides, you can appreciate these are the primary oocyte surrounded by the follicular cells, and in this case you can see the primary oocyte again surrounded by the granulosa cell, that is the single layer of simple cuboidal epithelium, and in this you can see the two layers of the simple epithelial cells of the granulosa, and here you can see the secondary follicles in which lichar folliculi containing spaces are present. So we will discuss all of the follicles one by one. The primordial follicle containing a primary oocyte. You can see over here there is a pool of the primordial follicle in the cortex, and out of these pools in every cycle few of the primordial follicles start grow and only one reaches the full maturity. So they are arrested in prophase stage of the meiosis 1. Their size is nearly 25 to 30 micron meter. Follicular cells are single layer of the flattened cells attached by the desmosome. You can see in this diagram, this is the lower magnification and this one is the higher magnification of the primordial follicular epithelial cells. Next, because of the effect of the follicle stimulating hormone, the follicular flat epithelial cells first become the cuboidal and then they become the stratified. And when these all changes are occurring, zona pellucida is also secreted and it is present between the primary oocyte and the surrounding granulosa cells. Also at this stage, stroma cells rearrange themselves around the growing follicle and they form the theca interna and theca externa. Then the secondary follicle develop, in which the size of the oocyte is nearly 125 to 150 micron meter in diameter. Antral spaces appear. You can appreciate the granulosa cells surrounded by the theca cells. The inner theca interna is cellular layer and outer theca externa is consists of the connective tissue fibers. With the growth of the follicle, the Graafian follicle is formed, still containing primary oocyte, which is arrested in the first meiotic division, and its first meiotic division will only be completed under the effect of the luteinizing hormone, which is released at mid-cycle. Continued growth of the size of the follicle, its size become 2.5 centimeter. Continued formation of the lichar folliculi. These are the spaces showing antrum which contain the lichar folliculi. So next is the ovulatory phase, that is the release of the mature Graafian follicles from the oocyte and its capture by the oviduct. Day 14 of menstrual cycle, there is a surge of the luteinizing hormone which causes rupture of the wall of the Graafian follicle under effect of prostaglandin, histamine, collagenases and hyaluronic acid. So at this stage, the primary oocyte completes its first meiotic division and starts the second meiotic division, but again arrested at metaphase 2 of the second meiotic division, and then it is released. So second meiotic division will only be completed if it is fertilized. Then is the third phase, that is luteal phase, in which residual follicular cells become part of the corpus luteum. So you can see in this diagram the Graafian follicle is extruded, ovulation has taken place, and remainder of the cells show luteinization, in which the granulosa cells become the granulosa lutein cells and theca cells become the theca lutein cells. Fate of corpus. What is the fate of the corpus luteum? It depends on whether the pregnancy occur or not. So if pregnancy does not occur, corpus luteum degenerate and it becomes corpus albicans. There is initially hemorrhage in the corpus luteum, causing the corpus luteum to become corpus hemorrhagicum. Later on fibrosis occur and corpus albicans form, and this carbon albican then degenerates and forms the corpus atreticum. But if pregnancy occur and there is the fertilization, there is continued release of human chorionic gonadotrophin hormone from the developing zygote, and this human chorionic gonadotrophin hormone maintains the corpus luteum, and corpus luteum becomes corpus luteum gravidarum or corpus luteum of pregnancy. So corpus albicans is because of menstruation. There is the corpus luteum is invaded by the fibroblast, and there are the fibrotic changes. In this diagram you can see these are the fibrotic changes in the corpus luteum. It is the remnant undergo the luteal, its remnants show the luteolysis, and fibrotic scar is formed. And this is the end of the 2 days slide. So we have studied different phases of the development of the Graafian follicle and phases of the ovarian cycle. So inshallah in the next class we will discuss about the histology of the uterine tube. Till then students, Allah Hafiz.